you guys get like I think so. Look, what's your guys' opinion? I have something about like spiking legs. Past that spike? Yeah. Was it that one? Five. Oh my gosh, we're six. You're five. Yeah. Oh, yeah.
All right. So today, we're going to continue talking about the male reproductive system. Okay. So when we left off last time, we had talked about the development of the reproductive tract in general. So we talked about how we have a generalized sort of indifferent form for a while at the beginning of development. And then in order to develop a typical male or female reproductive tract, we're really zooming in on, do we have the presence of a Y chromosome? In which case, we should have an SRY gene on it, which codes for testis determining factor, which causes the body to develop testes that then produce testosterone and Müllerian inhibiting hormone that causes our Wolffian ducts to develop and our Müllerian ducts to degenerate, giving us the male reproductive system. We talked about the fact that if we don't have those things, then we develop a female type reproductive tract. We had started zooming in on some of the portions of the male reproductive system, looking at some of the organs. I just pulled this picture forward. So we were about to start talking about sperm production. So I'm pointing out to you that as we are developing the sperm, we're going to have them developing in seminiferous tubules in the testes. We're going to move into a region called the rete testes, which then has efferent ductules in it, leading us into the epididymis. So as our sperm develop, that's where they're going. From that point, they are going to travel out the vas deferens, which swirls around. We're going kind of behind the bladder here. So we can see that we are now coming past the seminal vesicle as we pass through the prostate in the ejaculatory duct. So we're picking up some enzymes here. We're continuing through the prostate. We're picking up more stuff from the prostate. We're in the urethra, but as we pass by our bulbourethral gland, we pick up a final bit of sort of contribution to the semen as we pass through, and ultimately out the end or the glands of the penis. Okay, so highlighting those accessory glands, this is really where we left off, right? So those seminal vesicles here above the prostate have an alkaline fluid. They've got sugars, they've got enzymes, they've got prostaglandins. The prostate gland has more enzymes as well as citrate. And those bulbourethral glands, there they are, have a kind of viscous, mucousy fluid that they're adding to the contents of our semen here. Okay. This is just a table showing some of the selected actions of androgens like testosterone in the male reproductive system and on secondary sex characteristics. So you've got that there. This is just from your book. Now we're going to be thinking about what sperm look like and how they develop. Okay. So if we are looking at a sperm cell, we're going to see that it's kind of chunked into three pieces. Okay. So we're going to have a head up at the top here. It has both a nucleus. So this is where we'll have our chromosomes, our genetic information, remembering that this is going to be a haploid cell because it's a gamete. So it only has half the typical amount of genetic information there in that nucleus. We also have this front piece on the head called the acrosome. So the acrosome is like kind of a baggie of enzymes. So the enzymes in the acrosome are going to be needed for fertilization to occur. So this is going to kind of like break open and help fertilization when our sperm reaches an egg potentially. Then we have a mid piece to the sperm cell. So this mid piece is full of mitochondria. So we need mitochondria because we need ATP because sperm have to swim. So they need to produce a fair amount of energy so that the tailpiece, which is the third part, can make these whip-like movements that are going to allow the sperm to move. So for that purpose, we've got lots of mitochondria, so we can generate lots of ATP through aerobic cellular respiration there in the center. Okay. So a reminder before we go into the development of the sperm about where we kind of are in the testes. So this is a cross-section through a testis, as well as through the epididymis. Last time we had mentioned that we have these two different kind of cell types that we were interested in here. We had these Leydig cells, these purple ones on the outside, which were the kind of interstitial cells that are secreting testosterone. And lining the ducts of the seminiferous tubule, so lining these tubules, we have Sertoli cells, which are the target cells, so like the recipients of testosterone as well as FSH, and do a little secretion of their own, but what they're secreting is kind of an inhibiting hormone. Okay, so that's where we're going to find ourselves as we start to think about how we get sperm maturing and kind of depositing and collecting here in the lumen of the seminiferous tubules. So the other thing to note here is those tight junctions between the Sertoli cells that mean we have a kind of separate basal compartment and luminal compartment if we're looking at kind of the layers of this membrane. Okay. So what's going on with sperm is throughout the lifespan and can produce sperm, right, through the process of spermatogenesis. And the spermatogenesis is occurring really between the Sertoli cells. So we're kind of going like here between two cells. So here we see that tight junction there. Okay. So this can happen continuously because what we have up here at the top part, so we're on the outside at the top of this image. This is our basement membrane. So this is away from the lumen, so away from the center of the tube. We have our kind of like parent cells for this process, the spermatogonia. So you can see the spermatogonia up here at the top. Okay, so we have spermatogonia. They are actually diploid cells. So they are making more and more of themselves potentially. So they undergo mitosis, making duplicate copies. But we also have the ability for one of these spermatogonia to undergo meiosis and start to have those daughter cells of meiosis move down between the neighboring Sertoli cells. Okay. So as this happens, the process of mitosis just makes duplicate copies, but our goal for meiosis is to end up with four, ultimately spermatozoa that are haploid and that have different genetics from each other, right? Because kind of the goal of sexual reproduction is to mix up the gene pool, right? So we want to end up with people who are not all clones of each other. So these spermatozoa, we're going to have different genetics from each other. We're kind of parceling out pieces of the genome. Okay. So as we have the spermatogonia enter mitosis, we're going to leave one spermatogonium up here at top to just make more spermatogonia so that we can keep that sort of progenitor cell, that first cell up here at the top so we can make more sperm later. But here we have the other one entering meiosis 1. So we're going to next end up with two primary spermatocytes at this sort of entrance to meiosis 1 here. So this is getting towards our ultimate goal of having mature sperm cells. Okay, so we're entering meiosis. So we're going to start with these primary spermatocytes, and we can see that those primary spermatocytes are crossing actually through and past that tight junction. So this is a controlled process, but we allow these spermatocytes to go through. Okay. So meiosis continues. So after our primary spermatocytes, which are diploid, and we get two of them, we're going to move forward and get our secondary spermatocytes. We still just have two of them, because this is how meiosis works, but now they're haploid. So we've split it in half, so we no longer have pairs of chromosomes anymore. We just have one individual copy. in each of these two secondary spermatocytes as we're continuing down. Okay, so that's the product at the end of meiosis one. Okay. As we continue down towards the lumen of the seminiferous tubule, we divide again, so that's meiosis two. And at this point, we're pulling apart the sister chromatids, if you remember your terms for meiosis. So we've still got haploid cells, but we don't have as many kind of copies of each of those genes anymore. And now we have 4 cells because each of those secondary spermatocytes split in half. Okay, so those are our spermatids at the end of meiosis 2. So at this point, we've parceled out all of our genetic information here with the spermatids, but we're not totally done. So we still have to have some changes to the structure of those cells. So as we move down further towards the lumen, we're going to produce spermatozoa as these cells start to kind of like differentiate and develop those characteristic sections, right? So we're developing a separate head region, a separate mid-piece region, and that separate tail region as we're getting deposited into the lumen. So the genetic split happens first through our process of meiosis. Then we have our spermatozoa as those cells differentiate and take their more typical mature shape here as we get towards the lumen. So those are our spermatozoa. Okay. So this whole process that we were looking at there, this is called spermatogenesis. So genesis for creation, spermata for creation of sperm. So when we say it's happening from the basement membrane to the lumen, we mean it's happening from that outside layer to the inside layer where we have the opening for that tube. Okay. And as this is happening, we're making sure to leave one of those original spermatogonia up at top near that basement membrane up at the top, so that as it continues to reproduce over and over again, this is how we maintain the ability to produce sperm day to day and continuously through the lifespan. So after that process of division, so those different parts of meiosis, and then the differentiation of the spermatids into the spermatozoa, those spermatozoa are in the lumen of the seminiferous tubules, but they're not fully mature, which is why I was trying to avoid that word a little bit. So they have to stick around in the lumen of the seminiferous tubules for a little while. So they stay there for about 20 days. And during that time period, they can't move. So they're immotile for a fair while. So they're not going to become motile until they move really out of those seminiferous tubules and really out of the testes. So from the seminiferous tubules, we were taking, this is why I was pulling up that cross section of the testes beginning. We're going to be moving from the seminiferous tubules and the testes into the efferent ductules into the epididymis. This is happening by peristalsis and the flow, like so just like the actual movement of the fluid that's in the tubules. So we've got like kind of pulsing of kind of like little bits of smooth muscle. You can imagine that peristalsis process. I'm just going to imagine it as analogous to what's going on in the GI tract. And as they move into the epididymis, after this immotile period in the seminiferous tubules, that's when they're going to start to be able to move. So that's when they gain this ability to use their mitochondria for producing lots of ATP and whip those flagella, those tails, back and forth. Okay. From the epididymis, they move into the vas deferens, go up through the spermatocord, and then come eventually back down and out the penis and the urethra in a minute. So until ejaculation, like this process is going on, right? Kind of all the time, basically. So we're all the time going to be producing these spermatozoa. We're going to have them maturing. We're going to have them moving to the epididymis. They're going to kind of sit around in the vas deferens until ejaculation occurs. So they might be there for a little while. Which brings us to the sexual response, which is the next piece we would need for sexual reproduction to occur. Okay, so during the sexual response in males, the goal from a biological perspective is ultimately sexual reproduction, and we got a bunch of controls on this, right? So we're trying to get the sperm somewhere. So this is going to be under the control of the autonomic nervous system. So that relatively involuntary portion of the autonomous nervous system. And we're going to have some spinal reflexes involved here. And we're going to see kind of a switch over from which part of the autonomic nervous system is active and signaling as the sexual response continues. So when we start, we're going to go through like 3 phases for the sexual response. So the first thing that we need to happen, the first thing we're going for is erection. So the penis is going to go from flaccid to erect, so hardened, swelled with blood, basically, keeping that tissue straight. And this is happening as a reaction to the parasympathetic nervous system. So the parasympathetic nervous system is in charge of erection and that initial sexual arousal, basically. Then we're going to see a switch. So we've got this kind of middle phase as stuff starts to move. So after erection, we're going to see this switch over to the sympathetic nervous system during this emission phase. And this emission phase continues until we hit the point of ejaculation, which is also in control of the sympathetic nervous system. So parasympathetic is just the beginning, and then emission and ejaculation are taking us to the sympathetic nervous system. Okay. All right. In terms of stimuli for this process, we're going to be thinking both about brain function and about just like receptors, right? So we're going to have mechanoreceptors. So we're thinking touch receptors in the penis and in other erogenous zones. We're also going to have signals coming down from the higher brain centers from thoughts also involved in this process. So when we're thinking about stimuli for this switchover, that's what's happening. That's where we're creating this kind of switchover. Okay. So in terms of how this works in a book flowchart perspective, so we're going to start out with activity in descending neural pathways from the brain, aka thoughts, right? So we got thoughts coming from the brain here, as well as mechanical stimulation of those mechanoreceptors in the penis. So we've got thoughts and we've got feelings. We've got thoughts and touch here. All of this is going into the spinal cord. All right, so the spinal cord is what's going to be conveying these impulses out. And the first part that we're going to have as we are going towards our goal first of erection, right? That's the first stage, the first phase of the sexual response. So what we're going to be doing is we're going to see that there's an increase in activity in parasympathetic innervation to the penis at the same time as we have a drop in sympathetic activity. Okay. The goal of this is actually just to change the situation for the blood vessels, right? So erection is a filling and swelling with blood. So what's happening as we have this parasympathetic innervation rise is we're changing those arterioles that are giving blood to the erectile tissue in the penis. Okay, so what's happening? We got smooth muscle around those arterioles. The smooth muscle relaxes in response to the parasympathetic innervation. So as it relaxes, we know that means we're basically going to have dilation of those arterioles, right? They're going to get bigger, their diameter is going to get wider, which means we're seeing a drop in resistance, which means blood flow is increasing to the penis because of this influence of the parasympathetic nervous system. Okay. So then we've got extra blood flow going into the penis, into these erectile tissues, which means we have an increase in the blood volume and in the blood pressure here. And an interesting thing happens at this point, because as those erectile tissues swell and get more filled with blood, filled with pressure, the veins are also there in that same structure. Those veins are in the penis as well. And so as the erectile tissues expand, they basically block off the veins. So the result of that blocking off the veins is that all that blood that's flowing into the penis is not flowing back into your circulatory system. It's getting stuck there, basically, which is part of what maintains the erection, is what maintains that kind of pressure of blood on the walls of the erectile tissue. Okay. So that is the full sort of physiological process for what's going on during erection. So that's the parasympathetic part. There is a slightly off-color mnemonic for remembering the part of the autonomic nervous system that's responsible for this, which is point and shoot. Point is the erection and the parasympathetic nervous system. So if you are looking to remember this easily, that one's kind of helpful. So that's the parasympathetic part for the erection. Next, we move towards the emission phase. So at this point, we've got an erect penis. We've responded to some mechanical stimulation. We've responded to thought centers. We've swelled those erectile tissues with blood. Okay, so at this point, for emission to occur, what we have happening is an extended response to those mechanoreceptors and that mechanical stimulation. So this is basically continued touching, and this is where we see our neural activity shift from the parasympathetic nervous system towards the sympathetic control of our tissues, sympathetic control coming from the autonomic nervous system. Okay, so during this emission phase, so this kind of middle phase, what's going to happen is we're going to start to see basically contractions. So we're going to start to have like kind of squeezing, pulsing. So specifically, what's going to be contracting is our epididymis and the vas deferens first, right? So when we produced those spermatozoa and then the matured sperm, we moved from the seminiferous tubules out of the testes into the epididymis. And now we have sperm waiting in the vas deferens. So now during this emission phase, after we've achieved erection, we're going to start to have those tissues contract, which is going to push forward the sperm that are in that tube, the vas deferens. And side note, this is why a vasectomy works as birth control, right? You still produce sperm, but we just kind of block that tube. We block that tube, nothing goes anywhere, right? So we're going to see these contractions of the epididymis and the vas deferens, as well as contractions of some later parts of our ductwork, specifically the ejaculatory duct. So that portion we're passing through the prostate and passing past the, this part. Right here. So our ejaculatory duct. So passing through the prostate, passing through the seminal vesicle. Okay. Getting us forward. All right. So this is where we're picking up those secretions from the seminal vesicles and the prostate, because that's where we're located. Okay. And the result of this overall is that now, instead of having the sperm outside the body, essentially, because the vas deferens is outside the body, at least for a portion of it, we're going to be moving the sperm forward into the body, into the prostate. As we're passing through the prostate and past those seminal vesicles, we're picking up enzymes, we're picking up fluid, we're picking up citrate, and we're moving into the urethra, which is our final tube that's going to lead out of the body. Okay. So ejaculation is the third phase. So this is the final response. So as mechanical stimulation continues, we hit this point at some point. This is mediated by sympathetic activity. So basically your sympathetic part of the nervous system is just staying active until this point. And when it reaches a certain point, it stimulates contractions of smooth muscle in the urethra now. So we've got a kind of like seamless flow from that emission phase where we're contracting the epididymis, contracting the vas deferens, contracting the ejaculatory duct. We moved everything into the urethra, and now we're contracting the urethra. As we contract the urethra, part of how we're doing that is we are contracting both smooth muscle as well as some skeletal muscle at the base of the penis. And this is at the same time stimulating closure of the urethral sphincter. This is because what we want to happen is we want semen coming out the urethra. We don't want urine coming out. So we're basically taking that tube, that duct, right, that urethra that could carry either, and making sure that we're just carrying the one thing at this point. So that's also part of the sympathetic nervous system's job here during ejaculation. So the end result is that semen moves through the urethra and out of the body and kind of resets at this point. So as you go back and review this, and we'll do a little review as we compare our reproductive tracts and reproductive function in males and females. But you want to make sure that you're able to think about the hormones and what different cell types they match up with. You want to make sure that you know the pathway that we're going through as we send those sperm on their journey. We want to make sure that we understand how the parasympathetic and sympathetic nervous system are both involved in the sexual response. Okay. So we're going to break in and do the first part of our comparison to the female reproductive hormone, or reproductive system, I should say. And one of the things that we're going to be tracing through as we think about the female reproductive system, so we're going to have to think about cyclical variations in hormones, because we have a cycle in females in a way that we don't really see in the male reproductive system. And this is going to link up with what's going on in both the ovaries and the uterus, these two parts of the female reproductive system. Okay. So when we're talking about cyclic changes in hormonal activity, really we're talking about the menstrual cycle, okay? Now, the menstrual cycle and the influence of these hormones and the fact that they are controlling the development and release of the ova, of the eggs, means that in the female, we have restricted periods of fertility. So in the male reproductive system, so long as we've hit maturity and the system is up and going, males are basically fertile like all the time. We can kind of always be producing sperm. But in the female reproductive system, this is only happening at specific periods of time. So a female is only going to be fertile when we have ovulation occur, basically. So we need to mature that egg cell and release it in order for fertilization to happen. So that's one difference from the male reproductive system. Another difference is going to be that while in the male reproductive system, we kept one of those spermatogonia up at the top by the basement membrane in the seminiferous tubules, doing mitosis over and over again, maintaining gamete production throughout the lifespan. In the female reproductive system, that first part of meiosis actually happens before birth. So we've got a limited number of gametes we can produce, a limited number of egg cells that can be released, because they're hanging out there like kind of in stasis. We don't have this continual production happening. So a little bit of terminology as we start this. Okay. So what we are thinking about when we think about these primary reproductive organs here, we're thinking about the ovary. So we have that here in yellow, here in yellow. We're thinking about the uterus and connections to it. So here we can see basically the uterine tube or the fallopian tube leading from the ovary into the uterus. From the uterus, the bottom part of that is the cervix, which then leads to the vagina, which is the opening to the outside of the body. Okay. So those are the like big picture structures. We're going to be thinking about what's going on in an ovary, and specifically what's happening inside a follicle in the ovary. So the ovary is going to contain many follicles. So we can see these kind of circles that we're looking at. So those are our follicles. Okay? And we're going to see them change in size, change in the cells and structures that they contain. So the follicles aren't just one cell, they're like a collection of cells. And this is going to be happening over the course of the menstrual cycle, again and again. So these follicles are going to be within the ovary. Each of them contains one ovum. So if we're looking at this a little bit, this is our primordial follicle. So this is a kind of like immature follicle that we're starting out with. Here we can see an oocyte in the center. So this is going to be like one egg cell that we're going to mature. And here we can see an outer lining of follicle cells here on this primordial follicle. Okay. So as we trace this primordial follicle forward through time over the course of a 28-day cycle, we are going to see new cell types developing and new kind of regions and areas developing. Okay. So in this follicle, we're going to start out with an oocyte, the egg cell or the ovum. And we're going to also have some granulosa cells here. So those are one of our adjacent cells in this primordial follicle. And these are the cells that are going to be the target for estrogen and for follicle stimulating hormone, FSH. So the granulosa cells in that way are kind of analogous to the Sertoli cells that we saw in males, right? So they're surrounding our gamete, right? So that's one way in which they're similar. And the other way in which they're similar is that they're receiving signals. So the Sertoli cells were receiving testosterone signal and the FSH signal. The granulosa cells are receiving the estrogen signal and the FSH signal. They're going to respond to those signals from other parts of the body. So they're going to respond to this estrogen and FSH and secrete some of their own chemical messengers in turn. One thing they can make is something called inhibin. So we saw inhibin as well when we were looking at the Sertoli cells. This is a negative feedback response, looping back and turning off the FSH secretion at a certain point. Okay. Other things that the granulosa cells are doing. So just like the Sertoli cells, we thought about them as like support for the developing sperm cells. The granulosa cells are going to be transporting some nutrients to that developing oocyte inside. They're actually going to be secreting estrogens themselves as well. So they're both a target of estrogen and a secretor of some estrogens. Okay. So for our egg cells to develop, we likewise need to undergo meiosis to get from a diploid cell, so like just a normal cell with all those homologous pairs of chromosomes, and get ourselves to a haploid gamete cell. But the time scale for this is different than we see in males. Okay. So for oogenesis, so genesis creation, oo is our egg. We're going to go from our oogonia, so that progenitor cell, to our ova, those mature egg cells. Okay. But you can see that instead of splitting this up based on like location, so in males, this picture was just like a crosscut through two neighboring Sertoli cells. We've got a timeline here. So the process of gamete production in females actually starts during fetal development. So the place that you would see oogonia is actually not in any people that you see walking around in the world, unless you are looking at a pregnant person because these oogonia are undergoing mitosis and differentiation during fetal life. And this is when they prepare for the process of meiosis. So those oogonia are producing primary oocytes, just like the spermatogonia produced primary spermatocytes in the lead up to meiosis. But after they do that, after we make these primary oocytes, we're going to enter a period called meiotic arrest. So this double slash line is just like a pause button. So that production of the primary oocytes and this meiotic arrest, this pause button, all of this gets set during fetal life. Okay? So our oocytes aren't going to actually continue to develop until we lead up towards ovulation. So during meiotic arrest, you're going to be born, you're going to go through your childhood, you're going to hit puberty, and it's not until the cycling starts and ovulation starts that we're going to enter meiosis 1. So we're in stasis for a long time. Okay. So prior to ovulation, we're going to see meiosis 1 happening, creating a secondary oocyte and what we refer to as our first polar body. So unlike our male reproductive system, which creates like 4 functional sperm, like 4 gametes, we're really just kind of producing 1 mature egg here, and the extra material, so like the other side of the split, is the small, like kind of non-functional almost cell. So we're really going 1-to-1 down the development of the ovum here. Okay, so prior to ovulation, we're going to develop that primary oocyte into a secondary oocyte. And that's not until after fertilization that we're actually going to hit meiosis 2. So it's not until that secondary oocyte meets up with a sperm and is fertilized that we hit meiosis 2 for the full maturation of that cell, for that full meiosis 2 to happen, which is kind of crazy. Good. So we've got a menstrual cycle going on that's going to lead to the release, generally kind of 1 by 1, of our secondary oocytes is what we're going to be releasing, really. So we're going to see changes to the ovary, and we're going to see changes to the uterus on a roughly 28-day cycle. And we can kind of split up thinking about those 2 organs separately. So we're going to be thinking about an ovarian cycle, where we're tracing through what's happening in the ovary. And we're going to be thinking about a uterine cycle, where we're going to be tracing through what's happening in the uterus, because both of these are changing, but not quite in lockstep. Okay. This is going to be driven overall, the cycle, by changes in estrogen, so one of our hormones, and progesterone, one of our other hormones, during the course of a month. So we're going to start by thinking about the ovarian cycle. So we're going to split the ovarian cycle roughly in half, okay? So we are going to have our first half of the ovarian cycle in the follicular phase. So this is going to be day 1 to day 14. And we start counting at the first day of your period, actually. So this is like, the period isn't at the end, the period is at the beginning. So the follicular phase is going to incorporate menstruation at the beginning, as well as some non-menstrual days, which is kind of part of why we're tracing the ovarian cycle and the uterine cycle separately. Okay, so we're going from menstruation to ovulation during this follicular phase, as we see the growth of our primordial follicle maturing and ultimately sending out our oocyte as we burst open that follicle, and that's ovulation. The second part of the cycle is the luteal phase. So this is going from ovulation to just before menstruation, so the last day before menstruation begins. This is going to be another approximately 14 days. And this is going to be characterized by the presence of the corpus luteum, which is basically this follicle, after it bursts, turns into a gland for the luteal phase that ultimately disappears. So that's what's going to be happening during the luteal phase. So we're chopping our ovarian cycle roughly in half. So during the follicular phase, so this growing follicle part of our menstrual cycle, we're going to see what's happening is we're getting signals in the form of FSH, follicle stimulating hormone, should match up with what's going on. We are stimulating that follicle to develop, as well as estrogens that are telling that follicle to grow, get bigger, and develop some of these new structures that we see as we progress through this image. Okay. Now, while we're only going to be probably releasing one egg cell during ovulation, at the beginning of this follicular phase, we actually don't know which of the oocytes it is that we're going to release. So we're going to see actually a couple of follicles starting the growth from primordial follicle into these larger, more mature follicle types. So we got a couple candidates that are kind of in a race to see which one gets biggest fastest. And then it's going to biggest follicle is going to win and take over. So as we're moving through the follicular phase, that oocyte is growing. So it's growing bigger. And we have the cells surrounding it developing. So those granulosa cells that were our target cells for FSH and estrogen, as well as kind of providing nutritional support for that oocyte, they're replicating and kind of differentiating into some more cell types as our follicle gets bigger. So we are going to see something called the zona pellucida in the antrum form. So that's this kind of region in the middle. So this beige and this blue stuff. So the antrum is this blue part. This is like basically a sack of water, kind of. So that's what we're growing and is ultimately going to kind of burst and push during ovulation. So these are forming as we move through our follicular phase. So we have a couple of follicles all developing at once, all kind of receiving the signal from FSH and estrogen. They don't all grow as fast, just like if you plant a bunch of seedlings or some flowers, they don't all grow at exactly the same rate. So at a certain point, one follicle is going to become our dominant follicle. So it's going to be bigger. We're communicating with the whole rest of our system with these hormones that we're producing. So it's going to kind of end up suppressing the other follicles that were growing during that period. And all of the follicles, other than the biggest one, are going to kind of like regress and die away once we have a dominant follicle. Once we have a dominant follicle, it's going to continue to develop. So it got big, other ones died away. Now they're going to create this corona radiata, so this radiating crown, as we have a Graafian follicle, which is a mature follicle that's kind of ready for ovulation. So when we hit that point at ovulation, we are going to be kind of waiting for a signal. So there is going to be a signal for ovulation to occur. But when ovulation does occur, we're going to see that antrum and the follicle as a whole burst and release the egg cell. Okay. So for those hormones that are triggering ovulation, we want to think about kind of a complicated interplay of hormones, unfortunately. So the first thing that we will see happening is we're going to see a spike in something called gonadotropin releasing hormone, so GnRH. And it's going to cause a surge in another hormone called LH, or luteinizing hormone. Okay. So we're going to see a spike in one, spike in the other. And that luteinizing hormone is really the trigger for the ovulation. So luteinizing hormone tells that Graafian follicle, that dominant mature follicle, that it's time to ovulate. So that follicle is going to burst, release the ovule, which is then going to go into the fallopian tube and head towards the uterus, while the remnants of that follicle stay up there in the ovary and are going to turn into the corpus luteum, leading us into this luteal phase. So that's what the luteal phase is named for, as we have the remnants of this follicle turning basically into a gland. So during the luteal phase, that ruptured follicle has turned into the corpus luteum, which is a gland. Specifically, what that gland secretes are both estrogens and progesterone. So progesterone is going to be coming in here into play as we think about that corpus luteum. So it's going to be producing these hormones. And it's going to hit maximum activity about 10 days into the luteal phase. And then it's going to die off. So when it dies off or degenerates, that's this luteolysis process. And so now we're not producing those hormones anymore because this gland is gone away, it died off. So as we reach the end parts of the luteal phase, we're going to see decreased levels of estrogen and progesterone. So we're going to see a dip in these sex hormones. And it's these dip in hormones that's going to trigger menstruation. So it's going to ultimately be triggering a change in the uterus, causing the uterus to shed its lining and start us over in the follicular phase. Now, if fertilization does occur, so for fertilization to occur, we'd have ovulation. You've got about like 3-ish days where fertilization could occur. If it does occur, it would be right here somewhere at this beginning of the luteal phase. Then we'd see that corpus luteum be kind of like maintained, and we'd continue to produce hormones and start to produce new ones. So this is what happens. The cycle is what happens in the case that fertilization does not occur. Okay, so we'll pick up here on Wednesday. We'll see y'all then.